Hello everyone. This week we're going to talk about microscopes in the lab. So let's go ahead and get started. Microscopes are used to view microorganisms that are too small to be seen by the naked eye. So a brief history of microscopes. In 1665, Robert Hooke used handmade lenses so that he could see the details of insects and plants, and he was able to see the cells of these specimens. Now back then they didn't used to call them cells, they would call them blocks, and eventually those blocks are, what we refer to as cells now. In 1674, Antoine Van, he actually developed a more powerful set of lenses that were able to magnify the specimen up to 500 times. And of course, now our microscopes can magnify more than 500 times. And he was the first person to actually observe living single-celled organisms that he used to call back then animalcules. Microscopes designed by Hooke and Van, those are actually simple versions of the compound light microscope that we usually use now in the lab. So let's go ahead and take a look at a compound light microscope together and talk about how to use a microscope and talk about the parts of the microscope. Before we talk about the parts of the microscope, the compound light microscope that we use are usually parafocal. What does that mean that they are parafocal? It means that when one lens is in focus, all the other lenses are going to be in focus as well. And this makes things way easier for us when we go ahead and use that microscope in the lab. You go ahead and focus one of your lenses, and whenever you switch to another lens, your specimen is still going to be in focus. You might just want to fine-tune the focus, but you're still going to be able to see your specimen, which makes, as I said, things way easier. Now, the compound light microscope that we use usually has a set of objective lenses. The first objective lens that we're going to talk about is the 4x objective, and that one is called the scanning lens. We also have the 10x, and that one is called the low power lens. And in microbiology, we usually start with this lens just because the 4, you're not going to see anything because microbes are way too small, so you can actually skip using the 4x and use the 10x right away. Even though with the 10x, you're barely going to see any details about the microbe that you are looking at under the microscope, but it's a start. We also have the 40x, which we call the high power lens, and the high power lens, when you go ahead and use it, you never use the coarse knob. So I'm going to show you an image of the coarse knob right now, and you never use the coarse knob when you're using your 40x. You only use the fine focus knob. And the reason why you don't want to use the coarse knob, it's because when you use the coarse knob, you're going to notice that every time you rotate that coarse knob, your stage where you have your slide gets closer to the lens that you are using. And so what happens is when you use the 40x, you're going to see that the space between your lens and your slide, it's very, very minimal. So if you use the coarse knob, you actually might break your slides. And I'm going to show you a picture, can explain it a bit more, but with the 40x, never use the coarse knob. We also have the 100x, and this one is used heavily in a microbiology lab. So the 100x, we call it the oil immersion lens. And again, with the 100x, you never use the coarse knob. You can only use the fine focus knob. And again, same reason, you never want to use the coarse knob with the 100 or the 40 because you can break your slide. And with the 100x, you must use immersion oil. So we always put a drop of oil onto our slide. Now, by the way, the oil cannot touch the 4x, the 10, or the 40. If oil touches any of these three objectives right here, you're going to damage these objective lenses. So the only lens that can touch oil and get in contact with oil is the 100x, the oil immersion lens. Now, when you come and talk about total magnification, so you want to know the total magnification of your specimen, we go ahead and we calculate it by multiplying the power of our ocular lens and our objective lens. So the ocular lens, it's usually 10x. So it usually magnifies your image up to 10 times. Now, what do I say? Usually because sometimes an ocular lens can magnify your image up to 7 times, 15 times. So usually the magnification is going to be written on the ocular lens. So always take a look at your ocular lens and see what's the magnification power that's on that ocular lens that you are using. And then you go ahead and you take that and you multiply it by the objective lens that you are using. So if you're using the 40x objective, what's your total magnification? You're going to multiply 10 by 40, so your total magnification is going to be 400. If you're using your 4x, again, multiply 4 by 10, your total magnification is 40. So the total magnification for the scanning, it's going to be 40. The total magnification, if you're using the 10x, it's going to be 100. Total magnification for your high power lens is 400. Total magnification for the oil immersion lens would be 1000. Because again, you're going to multiply the 10 by 100. So the total magnification for the oil immersion lens is 1000. Let's talk about the parts of our microscope. So up here would be your ocular lens. So the eyepiece or the ocular lens. And basically this is what you look through at the top of the microscope. And as I said, typically your ocular lens, they have a magnifying power of 10x. And again, that number is going to be written right here on the side of your ocular lenses. So that's the ocular lens right here. Then we have the body. So this is the body of your microscope, and this is the arm. And so usually when you carry the microscope, you grab on the arm and you always put a hand on underneath your microscope on the base so that you can, ensure that you are carrying it properly and you're not going to drop it. This is the on and off switch. Now, it doesn't always have to be on the side. Sometimes it's on the side, sometimes it's at the back, but this is basically the power switch of your microscope. You also have a light intensity adjustment knob right here. Again, sometimes its location would vary, sometimes right here, and then sometimes you find it down here. So again, the light switch or the light adjustment knob can be found at various locations. And again, when you are viewing your specimen, you can actually adjust the light that's going to shine on your specimen. Now you're going to notice that the more you magnify your specimen, the light usually dims, and so therefore when you use a higher magnifying lens, you're going to notice that you're going to turn up the light, okay, intensity. Right here, we actually have those stage controls. Those knobs right here, when you rotate them, that stage can actually either move backwards and forwards, or it can move right and left. So you never manually move your stage. You always use the stage controls right here so that you can move your stage. And as I said, this is the base of your microscope. The light source is right here, okay? We have the set of our objective lenses. So you have your scanning, you have your low power, your high power, and your oil immersion lens. And they're all attached to this piece right here, which we call it the revolving nose piece, because you are able to rotate it. This is the stage where you're going to go ahead and place your slide on the stage. And on the stage, you're going to notice that there are those stage clips, and those stage clips, they usually ensure that your slide is placed in position and it's not gonna move around. So the stage clips kind of hold on to your slide. Now, when you want to move the stage, as I said, use the stage controls right here to move your stage right, left, backwards, forwards. You have the fine focus knob right here, and the fine focus knob, it's used to basically fine focus your image so that it's not blurry. So you go ahead and use the fine focus knob. Now the fine focus knob, you can use it with any of your lenses. So the fine focus knob, I can use it with my scanning, with my low power, high power, and oil immersion lenses, okay? This one right here, it's the coarse adjustment knob, and this is the one that you usually use so that you are able to see your specimen and view your specimen. So that's the first knob that we usually use. And when you rotate your coarse adjustment knob, you're going to notice that the stage can move up and down. So when we are trying to view our specimen, we usually rotate our coarse adjustment knob, and what happens is you keep bringing that stage higher up, closer basically to the lens. Now notice how when you use a lens that's, let's say for example 40x, okay, let's say this one is the 40x, notice how the space between your lens and your stage gets really, really narrow. And so whenever you are using the 40x or the oil immersion lens, never use the coarse knob. Because if you use the coarse knob and bring the stage any higher, the slide is going to be right here. If you bring that slide any higher up, closer to the lens, what's going to happen is your slide is going to break. Okay, so the coarse adjustment knob is to be used only with the scanning, the 4x, and the low power, the 10x. Never use the coarse adjustment knob with your high power, the 40x lens, or the 100x, which is the oil immersion lens. Never, again, use the coarse knob with the high power or the oil immersion lenses. There is the diaphragm down here below the stage, and the iris diaphragm, or you can just refer to it as the diaphragm, it basically controls the amount of light that's going to reach your specimen. And we also have the condenser. And the condenser has actually a lens right here. So the condenser, it's basically used to collect and focus the light from the illuminator onto the specimen, okay? And then we talked about the stage, and this is where you're going to place your specimen on it. Now, what are the steps that we take or follow when we are using a microscope? So the very first step is how to carry the microscope properly. So when you carry the microscope, make sure that you grab onto the arm of the microscope with one hand, and always go ahead and place your other hand to support the base of the microscope, just to make sure that you don't, lose your grip and drop the microscope, okay? So always put that other hand on the base. And then after you go ahead and you place it at your station, you go ahead and you turn on your power, okay? Your power switch, you go ahead and you turn it on. And then you go ahead and adjust the brightness using the light intensity knob. Now, as I said, you can adjust your brightness, but never make it too bright because it's going to hurt your eyes. Okay, now, so always kind of, dim the light. Now, if you need more light, of course, go ahead and adjust it. But again, don't, don't make it too bright. It's going to hurt your eyes. And then you go ahead and you clip your slide. So you place your slide on the stage and you go ahead and you clip it in between those stage clips that I just showed you. And you center your basically your slide over the light. Now, if you want to go ahead and center it over the light, what you should do is use the stage controls so that you can move your stage either backwards, forwards, right, or left, until your slide is right on top of the light source. Next, you should try to focus and bring your specimen to focus, and we do that by bringing the stage up using the coarse knob. Remember how I said the coarse knob can bring your stage up and down? So if you want to view the specimen, you want to keep rotating that coarse knob, and that's going to start raising your stage. You keep raising it, okay, as you're looking through the ocular lens, and then you stop raising your stage, once you see something through that ocular lens. Once you see your specimen, you stop raising your stage, and then you go ahead and you fine focus your image. And again, just a quick reminder, the coarse focus knob is to only be used with the 10x objective, okay? Or the 4x. The reason why I didn't mention the 4x, because as I said, with microbes, they're super small, so we tend to even skip using the 4x and we move on to the 10x right away, okay? And then you move on from the 10x, you switch your lenses to the 40x so that you can see more detail, okay? Even with the 40x, you're barely going to, you're going to see your microbes and everything. It's just you're not going to see too much detail, so you will have to magnify even further. And with the 40x, never use the coarse knob. Okay, so with the 40x, that's it. Can't use the coarse knob anymore. You can only fine focus your image. Okay, so use the fine focus knob so that you can sharpen your image. After you fine focus your image, you're going to have to move on to the 100x objective. Okay, now with the 100x, you're going to put a drop of oil onto your slide, and then you're going to move the 100x so that it makes contact with the oil. Okay, so you put a drop of oil onto your slide, and then you go ahead and you click your rotate your 100x objective into position. It's going to touch the oil, which is absolutely fine. The oil can touch the 100x objective, but never let the oil touch any of your other lenses. If oil touches anything, any other objective lens, it is going to damage it. Okay, so the oil, basically, why do we use oil? Because oil is actually going to prevent the light from refracting, from bending. Okay, so we use the oil to prevent the light from refracting when we're using the 100x objective. Okay, otherwise, if the light basically bends, the image is going to be very blurry. Okay, so to prevent that, you must put a drop of oil when you're using your oil immersion lens. You can fine focus. Okay, so when you are using the oil immersion lens, you can fine focus. Never use the coarse knob though. Okay, but you can go ahead and use the fine focus knob so that you can sharpen your image. Okay. Now, when you're done viewing your slide, go ahead, switch to the 4x lens, lower your stage to the lowest position, remove your slide. Don't forget your slide on the stage. Make sure that you remove the slide. Wet a lens paper with some lens cleaner and go ahead and wipe your lenses. But by the way, never use the same lens paper to clean all of your lenses, because if you use that lens paper to wipe off the oil from your 100x objective and then use that same lens paper to clean your other objective lenses, what you're going to end up doing is you're going to expose your other lenses to the oil, and by doing that, you would damage the microscope. So make sure that you do not use that same lens paper, okay? Especially the one that you've used to clean off the oil off of your 100x objective. Never use anything other than lens paper, okay guys? So the lens paper is the only thing that you can use to clean your lenses. Anything else is going to scratch the lens and damage it, okay? So never use tissue paper, never use paper towels, cotton, none of that, okay? Only lens paper to prevent, as I said, scratching your lenses. And then to store your microscope, make sure to lower the brightness to the minimum using the light intensity knob. Make sure that you turn off the power of your microscope. Make sure that you place the 4x objective over the stage, okay? Wrap the cord around the back of your base. If your microscope has, a cover, go ahead and cover it up and put your microscope in its designated location. Now, right here, we're just going to introduce some of the morphologies or shapes of the bacteria and their arrangement. And we're going to see multiple pictures later on, but let's go ahead and kind of introduce some of the shapes that you're going to see. So, bacilli, it's the plural of the word bacillus, okay? What's bacillus? Bacillus means a rod, okay? So bacillus, it's a rod-shaped bacteria, as you can see right here, okay? So a rod-shaped bacteria, we refer to it as bacillus. Now, some bacteria out there, they are bacillus, and they are single bacillus, Okay, so when the way they are arranged, each bacillus bacteria is on its own. They are not touching. Okay, but some of them, as you can see, the way they are arranged, they are touching each other and they're like paired. Okay, so what do we call them? We call them diplo. Okay, so diplo coming from the word di, two. And then we say bacilli. So bacilli, it's the plural of bacillus. Now, could you have a bacteria arranged as a single bacillus on one day, and then on the other day, when you come and view it under the microscope, you would see it as diplo? No. Okay, you will never see that. So bacteria, they tend to stick with one shape and one arrangement. Okay, so if a bacteria out there is a diplobacilli, so it looks like a rod and they are paired, it's always going to look like a diplobacilli. Okay, so anytime you go ahead and you view that specific bacteria, it's always going to look the same under the microscope. Okay, it's not going to look like a diplobacilli today, but then a week later look like palisades. Absolutely not. Okay, guys? What is palisades? Palisades, those bacteria, they are rods, okay? So they are bacilli, but the way they are arranged, okay, guys, it's kind of like a picket fence. And so we call that arrangement palisades. So notice how so far the shape that I'm talking about, the shape is a bacillus. Okay, now how is those bacilli bacteria arranged? They could be single, they could be diplo, kind of paired like that. They could be arranged parallel like this, like a picket fence, and we call it palisades, or they could be in a chain. They could be arranged in a chain. When they are arranged in a chain, what do we call that? We call that strepto. So if you ever see the word, the prefix strepto, strepto meaning those bacilli are arranged in a chain-like format. So I'm going to call them streptobacilli. If they are arranged by, like, as twos, we call them diplo. Singular, just bacillus. Picket fence look, palisades. Okay. Now, another shape is coccus. Okay, coccus, it's spherical. Okay, what's the plural for the word coccus? Cocci. Okay, so cocci, it's the plural of the word coccus. And coccus, it's the spherical shape. Now, will you ever see a bacteria that looks coccus on one day and on the other day it will turn itself into a bacillus? Absolutely not. Okay, so bacteria, they stick with the same shape. Okay, so they don't change their shape. Okay, you guys? For example, E. coli, the bacteria E. coli, it's bacillus. Okay, so E. coli, it's always going to look bacillus under the microscope. Will I ever see an E. coli, changing shape and looking spherical, looking like a coccus? Absolutely not. Okay. A bacteria that's coccus in shape, it's Staphylococcus aureus. Okay, so Staphylococcus aureus will always look like a coccus under the microscope. Now, why is it called Staphylococcus? Because the way it's arranged, it's staphylo. What does staphylo mean? Staphylo means it's arranged in clumps, okay, kind of like in clusters. So the word staphylo meaning clusters, okay. So right here, staphylo meaning if your coccus bacteria, the way they are arranged are in clusters. You use the prefix diplo if your coccibacteria are arranged like in twos, pairs. Okay? Strepto, a chain. Okay. Tetrid, if your coccus bacteria are arranged 4, like, you know, 4 are stuck together. Okay, that's called tetrid. Now, can you ever have a bacillus that are arranged in tetrads? No. Okay, so tetrads, it's only if your bacteria are coccus in shape. Strepto, yeah, you can have streptococci, okay, and you can have streptobacilli, okay. Staphylo, it's always cocci. You don't see staphylobacilli. That's not, it's not found, okay. Diplo, yes, you can have diplococci and you can have diplobacilli. Okay. Singular, yeah, you can have a singular coccus on its own and you can have a singular bacillus on its own. Okay. Palisades, it's palisades, that's an arrangement for bacilli. You don't see palisades for cocci. Okay, guys, so again, there are some arrangements that are common between the two shapes, okay? And then there are some arrangements that are only specific for coccus bacteria or for bacilli bacteria, okay? And then we have Spirolia. So some bacteria out there, they kind of look like a common shaped, or we call it Vibrio, okay? Kind of reminds me of an elbow noodle, okay? So they kind of look like an elbow noodle and we call them Vibrio. Some of them are, they look something like this, like a snake, and we call them Spirilium. Some of them are thinner and they're more even spirally. We call them spirochete. Okay, so those are some shapes of bacteria that you need to be familiar with. So you need to know the shape and then you need to know what type of arrangements they might be arranged in. Okay. And then those are, right here you have a video to watch that will go over how to properly use a microscope. There is a virtual lab for you to try out as well. And then right here, you do have some images that I do want you guys to go ahead and take a look at. And those images will help you out to answer certain questions on week 3 worksheet. If you want to zoom in on these images, go for it so that you can go ahead and get a better view.